previously we were together in lecture one. I believe you are now well acquainted with what we covered in lecture one. This is the second lecture, and we are going to look at fetal growth and development. So, by the end of this session, you people should be able to now explain the process of fertilization, identify the stages of fetal development, and describe the embryonic and fetal structures. So, what is fertilization? So, when we talk about fertilization, we basically mean that union between the spermatozoa of the male, then the egg or the ovum of the female. So, in this case, it involves ovulation, which is the release of the ovum. Then also we have release of the spermatozoa, which happens in most cases through intercourse. Then this will travel to the fallopian tube, and specifically at the antler where fertilization will occur. So the viable period of the sperm in the female reproductive system is about 24 to 72 hours. So in case the sperms are released into the female reproductive system and they do not meet the egg or the ovum immediately, they can stay alive for up to 2 to 3 days. That is from 1 up to 3 days. That is from 24 hours to 72 hours when they are still alive. But after the 72 hours, they will not be viable. So right there we have the structure of the female reproductive system illustrating the sperms swimming through the reproductive system until they meet the antler. Though they are directed towards that the left ovary, that is because this space, the release of the egg at that month happened from that side. That is why the sperms are directed that side. But when they are released, they run randomly to both sides. So let's look at the alkaline cervical mucus. So alkaline cervical mucus is it refers to a secretion which is produced by the cervix of the female reproductive system. So this we know that the vaginal fluid is somehow acidic in nature. But again, when the woman is nearing or is within her danger days, or what we can refer to as the days in which she can be able to conceive, the cervix produces uh, mucus or fluid which is alkaline in nature, and this is the one that allows for the smooth passage of the spermatozoa until they swim into the uh, fallopian tube. So this mucus produces a suitable environment to make sure that as the spermatozoa swims through the reproductive system to reach into the fallopian tubes, they are not injured, they are not harmed, they move in a safe environment. The acidic environment is dangerous to the spermatozoa, and in most cases it can be, in case the environment is acidic, it can be one of the reasons that leads to infertility in some couples. So we still continue with fertilization. So basically, like I told you, is the union or the fusion of the spermatozoa with the ovum or the ovum's nucleus. So there is chemical change that happens immediately when the spermatozoa meets with the ovum. So once the first spermatozoa meets with the ovum, there's what is called zonal pellucida, which is released. This is an enzyme which is released by the egg, and this is to make sure that it seals the egg off from these other spermatozoans. That's it. are swimming towards the egg. So once the first sperm touches the egg, then the zonal pellucida is released, and all the other sperms are shielded off from the egg. So make sure that uh, it remains that way, because that is the natural way of um, of how fertilization should take place. Otherwise, it may happen that when the egg is fertilized with more than one sperm, which can happen in some cases, then it can lead to multiple pregnancy or pregnancies that are more than even two. So that can be explained when we are talking about the abnormal pregnancy. So currently, as we are focusing on normal, we shall only focus on what is normal. So the breakdown of spermatozoon's membrane occurs also during this time, and there is formation of a naked male pronucleus. So in this case, the the, the egg is already fertilized, and it is what is referred to as zygote. So right there we have that picture here, which is showing the how fertilization takes place. So as you can see, once the first spermatozoon has touched the nucleus of the egg, the zona pellucida is formed and shields off the other sperm to make sure that they don't fertilize the egg again. Deciduous in early pregnancy. So in early pregnancy, we talk about uh, um, the 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 walls of the uterus. So before the walls of the uterus, or before the fertilization takes place, the walls of the uterus are referred to as the endometrium. That is the inner walls of the uterus. So once fertilization takes place, or in that preparation, as the uterus is preparing to receive the fertilized egg, to receive the zygote, there's a lot of nutrients that are deposited in the endometrium, and as a result, it becomes spongy with a lot of blood circulation, and in that case, its name changes from endometrium to decidua. So let us look at capacitation. Capacitation is the activation process that prepares the sperm for fertilizing the egg, and begins once the sperm enters the upper female genital tract or uterus. So capacitation is like the process of giving energy for the sperm to fertilize the egg. So capacitation involves biochemical and biophysical changes in the sperm membrane, such as cholesterol efflux, increased membrane fluidity, protein phosphorylation, and ion channel modulation. So all these are changes that occur to make sure that the sperm gets energy to make sure that it can be able to fertilize the egg. There will be that outlet of the cholesterol from the sperm, and to make sure that the viscosity, or what we refer to as the membrane fluidity, is increased to enable it to fertilize the egg. Capacitation enables the sperm to undergo the acrosome reaction, which is an essential process of fertilization, and is characterized by the exocytosis of the acrosomal content, the contents that are the acrosome of the spermatozoa are let out, and there is also release of hybrid membrane vesicles formed by patterns of the outer acrosomal membrane and the plasma membrane. So right here we have we are having a pictorial which is demonstrating the acrosome reaction, how exactly it takes place. So you can see right there we are having um, the inner acrosomal membrane which dissolves, and as a result we have the secondary oocytes in the second meiotic division, and the other structures that you can take time and study them properly. So the acrosome reaction is a distinctive exocytotic process that is initiated by multiple vision events and the subsequent vesiculation occurring between the outer acrosomal membrane and the overlying plasma membrane. So it occurs between the two membranes. So the acrosome reaction exposes the inner acrosomal membrane, which contains proteins involved in the sperm-egg interaction, such as the zona pellucida binding proteins and the ulema binding proteins. So this is what actually takes place. So the acrosome reaction also modifies the sperm function. Since acrosome reaction are reactive sperm are no longer able to bind to the zona pellucida, but the ulema. So the ulema is that part of the cyst which is the which is uh, which the other sperm can reach, but they cannot now penetrate inside into the nucleus of the ovum. So the key stages in development include the following. There are three major key stages, and these are the pre-embryo stage, which is between 1 to 2 weeks, then the embryonic period, which is 3 to 8 weeks, or what is called organogenesis, then the fetal period, which is 9 weeks to full term. This allows further growth and differentiation of the cells. So let us look at the pre-implantation stage. This involves completion of blastocyst division, then arrival at the residua of the uterus and zona pellucida disappears. So when we talk about the blastocyst division, when the ovum is fertilized, it divides, it starts dividing, but before it divides, what is formed is the one that is called what is formed is the one that is called the zygote. Then after that, we shall look into how a cleavage is formed. Then it divides into blastostomere. Then it divides into morula. Then after that, now the blastocyst is formed. So that complete process is completed within the pre-implantation stage. Then after that, the zygote now moves into the endometrium. It moves the uh, from the fallopian tube or the ampulla where the fertilization has taken place. Then it moves up to the situa. The previously known as endometrium is now known as the situa because of the increased supply of nutrients and the uh, red blood cells or the blood specifically. So then from there, the zona pellucida, which was formed to shield off the other sperms from uh, fertilizing the egg, now will end up uh, it will end up disappearing. So let us look at early development. So in early development, rapid mitotic cell division occurs, and this occurs within the zona pellucida, which starts immediately within 24 hours after fertilization has taken So from there is the cleavage, which is formed. A cleavage is like a division which is formed within the fertilized ovum. So when that division is formed, it divides the fertilized ovum into two parts, and this is what we refer to as blastosomia. And these blastosomias also keep on developing until they give rise to what is called the morula. So zygote moves to uterine cavity with the periodical cyst and with ciliary motion. Then it reaches the uterus as a solid mass of uniform cells. That is the morula. So in most cases, they can be 16 to 50 divisions that occur. So right here, we have a pictorial which is demonstrating that. As you can see, this is what we call a cleavage right there. This was one fertilized ovum. Then there's this division that occurred, and it divided it into two. Then the two furthermore divided to form four. Then from there they kept on dividing until they formed the morula. Subdivisions of cleavage. So stage of compaction. Well, at first there's a stage of compaction. Then after that there's tight junctions that happen between the cells. Then from that it keeps on dividing like we have seen in the picture until they reach up to around 16 or more cells. That is normally reached at around the fourth stage. So fourth division is covered by zona pellucida, and this consists of inner and the outer cell layers. So the outer cell layers are the ones that are known as the trophoblasts, whereas the inner cell layers are known as embryo proper, that is also called the embryoblast. So the pellucida prevents implantation in the wrong site. So after that, we have the blastocyst, which contains up to around 32 to 64 cells, and the fluid passes through morula from the uterine cavity. The blastocyst attaches to the endometrium, and this is what we refer to as implantation, and it happens between the 6th up to the 12th day of after fertilization. So during this period, sometimes there is the spotting of blood that may happen, but it does not take long. So after implantation, it becomes the implanted zygote now becomes the embryo. So this is spotting in most cases can take like 1 to 2 days, then it clears off. So subdivisions of cleavage, you can see here. This is the picture that depicts what we have been talking about. We have the polar trypoblast, we have the inner cell mass, then the cavity of blastocyst, then trophoblast. Here also the same, we have already talked about it. So let us look at the inner cell mass or ICM. So it is a group of cells that form inside the blastocyst during early embryonic development. So the ICM is the source of embryonic stem cells. The ICM also gives rise to the three germ layers, ectoderm, and endoderm, which will form all the tissues and the organs of the embryo. So the ICM is surrounded by the protectoderm, which is a layer of cells that will form the placenta and the alga extra-embryonic structures. The ICM is separated from the protectoderm by a fluid-filled cavity which is called the blastocells. The ICM is attached to the protectoderm by a structure which is known as the polar body, which is derived from the first meiotic division of the oocyst. So let us look at the epiblast. The epiblast is the layer of cells that faces the trophoblast, which is the outer shell of the blastocyst. The hypoblast is the layer of cells which faces the blastocyst, which is the fluid-filled cavity inside the blastocyst. So the epiblast gives rise to the three primary germ cells, which are the ectoderm, mesoderm, and the endoderm, as I mentioned previously. And this normally occurs during gastrulation, as well as the amniotic ectoderm and the extraembryonic mesoderm. So we still continue. The hypoblast gives rise to the yolk sac and the chorion. And these are very, very important as far as provision of nutrients to the growing embryo of plant. Then the epiblast cells are pluripotent, meaning they can differentiate into any type of cells in the body. So let us look at the hypoblast. The hypoblast cells are not pluripotent. That means that they cannot differentiate into any cell of the body, but they play a role in inducing the formation of the primitive stress, which is a linear band of cells that marks the beginning of vasculation. Vasculation, we have talked about it. Let us get to know what it means. Is the stage in embryonic development when the two-layered structure, that is the epiblast and the hypoblast, become a three-layered structure, which is the ectoderm, mesoderm, and the endoderm. Let us look at germ layers. The embryo proper forms a three-layered germ disk consisting of ectoderm, mesoderm, and the endoderm. Which are the three primary germ layers? That when we talk about germ layers, these are layers that have potential of differentiating or developing into something else, differentiating into different cells, which is what we refer to as germ layers. They have that ability of developing. So they give rise to different types of tissues and organs in the body. So right there we have the germ layer that we talked about. We have the inner solid layer that covers the surface of the embryo. It forms the nervous system, the sensory organs, the skin, the hair, the nerve, the nails, and the tooth enamel. So it also gives rise to some endocrine gland. Then we have the mesoderm, the bones, the cartilage, the blood, the lymphatic system, the cardiovascular system, the urinary system, and most of the reproductive system. It also gives rise to some endocrine glands such as the adrenal cortex, the gonad. Then we have the endoderm, which is the innermost layer that lines the primitive gut of the embryo. It forms the digestive system, the respiratory system, the liver, the pancreas, the thyroid, the parathyroid, and the thymus. So it also gives rise to some endocrine glands such as the posterior pituitary gland. So those do not need much explanation. That's why we just pass it through because they are self-explanatory. So trophoblast is the outer layer of the blastocyst that forms during the early stages of development. Trophoblast has two main functions. One is providing nutrients to the developing embryo and facilitate implantation into the uterine wall, which we said is this time considered the endometrium, meaning the tubule. Then trophoblast consists of two types of cells, that is the villus cytotrophoblast and the extravilus cytotrophoblast. So the villus cytotrophoblast cells proliferate and diffuse to form the cytotrophoblast, which covers the surface of the placenta and is in direct contact with the maternal blood. Then the extravilus cytotrophoblast cells which migrate out of the placenta and invade the maternal tissues such as the endometrium, the myometrium, the spiral arteries. So these are the ones that can do this one. Trophoblast inversion is regulated by new molecular and cellular interactions between the trophoblast and the maternal microenvironment, including paracrine and endocrine factors. The trophoblasts improve endometrial susceptibility to the implanting to make sure that it attaches itself firmly, but again, not deeply into the myometrium. Once they reach into the myometrium, that is when we have abnormal cases of uh, uh, over anchoring of the placenta, which can lead to complications during delivery. The placenta cannot be delivered out because they are attached to the muscles. So let us talk about implantation, which happens between day 6 to 12. So implantation will is from day 6 up to day 12. There is the disappearance of zona pellucida, then readiness for embedding in the stigma. Then there is also formation of the body stalk connecting the inner cell mass, the trophoblast. Then there is also uterine lining, um, which prepares for the implantation. Then there's also formation of bilaminar disc with the epiblast, that is the amnion, and the hypoblast, which is the yolk sac. So let's look at the studio in early pregnancy. So the studio in the is the term used to describe the modified endometrium or the mucosal lining of the uterus. This I've already explained about it. So the visualization involves changes in the structure, the formation of endometrial stromal cells, and their interactions with other cell types, such as immune cells and blood cells. This is under the influence of X. So the stigma plays essential roles in protecting the embryo from maternal immune cells, providing nutritional support and participating in the formation of placenta or the stigma has several distinct parts, and this is the decidua basalis, decidua papularis, and the decidua parietalis. So those are the parts that are formed when the endometrium is transformed into the decidua. All those distinguished parts are formed. So the decidua bacillus is the part of the decidua that is related to the chorion. The chorion is the outermost fetal membrane, the outermost. That's when someone does vaginal examination, it's the first membrane that the person touches, and it becomes the maternal component of the placenta. So the sidua capsularis is the part of the figure that overlies the embryo and covers it from the uterine fluid. So right there we have images that are showing the embryo in early pregnancy. We have structures there like the placenta, the umbilical cord, amniotic cavity, amnion, chorion, and many other structures that we can talk about, including the different parts of the placenta that we have mentioned before. Then decidua. Hello? This is the rest of the decidual tissue that lines the uterus. The decidua undergoes a process called decidualization for it to become decidual. Remember, before I said that this is the part of the uterus that is known as the endometrium. So hormones such as progesterone regulate decidualization and the signals from the embryo, such as human chorionic gonadotropin, which is XCG. Decidualization can be impaired by genetic, environmental, or epigenetic factors, which may lead to early pregnancy loss or other complications. So let us look at the chorionic villi and the placenta. 11th to 12th day, chorionic villi will be formed. So chorionic villi are the projections of the chorion that provide maximum contact area with maternal blood for nutrients and the gas exchange to make sure that the embryo is able to feed and get some the blood and the nutrients at the same time. So chorionic villi are essential for the development of the placenta. Chorionic villi develop from the trophoblast cells of the blastocyst after implementation of the uterine wall. So right there, these are the structures that we are talking about. This structure shows the chorionic villi and the placenta. So right there we have this demonstrated further. Structure A shows primary villus, then secondary villus, then tertiary villus, which is now well developed. Chorionic villus and placenta continued. Primary villi are non-vascular and consist of only trophoblast cells, and they appear around 13 to 15 days of gestation. Then the secondary are larger and branched, containing mesoderm cells and trophoblast cells. They appear around 16 to 20 days of gestation. Then tertiary villi are vascularized and contain blood vessels derived from the umbilical cord. They appear around 17 to 22nd day of gestation. So thank you so much. That marks the end of our lecture for today. Very grateful.